Okay, welcome to chemical biology class. This is the first class of we talk about protein purification. Okay, let's start. So the cells, or the bacteria or mammalian cells, which is our cells, has thousands of protein. Thousands of proteins in the cells. And then the protein purification is basically we pick only one protein of interest. So say if the cells are producing pharmaceutically important protein, for example, insulin or growth hormone, and many others, EPO, erythropoietins, and many other things, their cells are secreting and producing in a large quantity. And then inside of this proteins are in here, we want to pinpoint and purify the protein of interest we want to purify. Also, the different case that cells are not overproducing particularly important proteins that we want, but generally speaking, in biologists, especially cell biologists, want to study the protein-to-protein interactions, which we providing the signals from the outside or the inside that all the signals are communicating through this protein-to-protein interactions. So the biologists want to know some signaling networks through the protein-protein interaction, so that the most important technique is to purify this protein, so that we can, from this, we can figuring out which proteins are connecting to each other. So protein purification in chemical biology is the most important technique to learn and understanding the basics of the chemical biology. So we want to talk about some protein purifications. So this is introductions. So then I want to give some examples, so which is not a scientific examples, but I want to providing such examples. If this is a real class, I give you some little bit simple game that if this is an individual student, for example, say we have 20 different students are sitting here, and then let's say we have 50% male, 50% female. So that maybe 3, 4, 5, 6, 7, 8, 9, 10. These are, say, the double circles are female, and then just a simple blue is a male students, for example. And also, if the male students, maybe they have some no hair, bald, for example. So this is a bald person. And then also the male students, among the male students, they have a shirt also. I'll say shirt. And then some are, in a both male and female, maybe they wear the jean pants. So I will say jean. So jean here, J and J, and here. Also, some have glasses, no matter what the male and females are. So I will put some glass, G here, G and here. So this is kind of examples that characterizing by the appearance of the individual students, for example. So this is providing a good example to how we making a strategy to purify the proteins. Similar, so this is kind of good examples that explain how we make protein purifications out of it. So if these peoples are representing one individual proteins, which has a different charge, different size is different, shape is different, they have all the different appearance in the case of the protein. Also, the human is same. Our individual students are having different appearance, I should say. So I will do like this. Say, I want to purify, say, raising the hand, which individual, all students are raising hand. If I call some category of this, then the students will put down their hand, for example. So raising all the hand, you can imagine the situations. Now, say, put your hand down for the female students. So female students are all hand down, so they're not purifying. So the step one is females are taking out. So the female out. So I did step one. So all the students are now clearly, I should say, removed from this list. So they are left over, right? And then if I say, say students with the jean pants is excluded, then I will delete it. I will say the step two and delete it out. Then I will say this two, this one, this one, delete it out. Right? And then if I say want to purify, well in this case protein by the students, but this is the one has a glass, then I will say remain an individual with glasses. Then automatically all the students are removed so that I can purify this one. So this is kind of a strategy that even if the thousands of different proteins are there, or 10,000, 20,000, no matter what there are, how many, but we can have characterized like this, some appearance way that this is a good example that I explained here. And we can take this, some populations are completely out, and then remaining things we can get those things. And then some part of the protein which is individual that completely out, and then we take the rest of it, and then repeating this process or different techniques to apply, then we can actually have the final protein species that we want. So this is it's simply difficult questions, and then 10,000 or thousands different proteins, how we can separate a protein, particularly interesting protein that we want to purify. But it's examples that can excluding some certain categories out of it, then we can actually purify successfully in a easier way to using the less steps of the process than you can imagine. Then you can actually purify the protein that we want. I hope that this is kind of providing a good example to us, to you, and how we make a strategy of the protein separations. So let's give real situations now here. So before going this way, I want to introduce the principles and sides so that we cite exclusion chromatography. So the other word is gel permeation, chromatography, so that we call this two different words, but SEC, site exclusion chromatography, is a more popular way to call this technique. The reason why we take this I explain the site exclusion chromatography first is related to our previous class, which is the hydrogen, because this technique basically has the following process, which has you have a bead. We take the beads, which is the size of the micron size bead. We take here. So we take the beads, putting in this long and big columns here, and then we put the water, and then this powder, initially powder, is swelled. It's called swelling. The swelling process. So that like imagine situations that then you eat rice, for example, and the rice is dried out, and then the volume is shrink compared to the initial process. The rice volume is like this way, then when this dry out, which is without water, the overall volume is shrink, right? But if you add the water back, then this dried individual rice particles is swelled, right? It called the swelling process. And then it becomes actually bigger and bigger, much bigger than the original size. So similarly here, so we have processed polysaccharides. This is the beads is basically polysaccharides or synthetic polymers. In both you can use it. And then this is dry powder, but as soon as we add water, then they become swelled. So they get in overall volumes are become bigger. Similar to the process that I explained in the examples as rice particles. And then they become to feel like huge fillings like this. So I brought actually the real examples to explain better for you to understand the system. So I brought the Cefadex G75, which is a commercially available the beads, which is you will see the powder, micron powder. So I will demonstrate this. So we can take a powder seed here. I will probably maybe you can see the height, right? So overall height is probably, for me case is about say 1 or 2 millimeters, for example. Then this powder, into which I will put the water out of it. So you can, here's water, so I can put here. Water's out of it. And then I make a little shake, which is like the particles of rice you can imagine. So then by the stirring, shaking of this, I can provide more water to this bead, so it can swell much faster. Then instead of that, just put the water and then remain sitting here. And then you will see in a minute that these particles are swelled, and then they become heavier and heavier as the water molecules go into the bead. They settle down slowly. So individual particles just goes in down, so they pack, and making hydrogel-like layers you will see. So I'll put this in a minute you can see how they look like. So then you'll see a little bit later. So then you see, I'll continue this explanations that the microbeads that you showed before is initially powder, and then I put the water or different solutions as you depending on the situations of protein purifications. And they becomes getting water and swelled, becomes volumes are much larger than the initial volumes of the beads. They can put something like this bead volume. So basically, this situations I can describe in which the individual beads are packing like this way, really well packing to this way. Okay, and then your proteins. And now the proteins are not a single type of protein. You have a mixtures of protein that you want to purify. So the proteins are heterogeneous proteins are going inside. Okay, so this beads is now separating the proteins as their size. This is basically, don't forget the size exclusion chromatography. So how they exclude. So that means excluding the size of the protein. So that I will explain how they providing the size. So now in here you actually see. Remember the initial volume is actually 1 or 2 millimeters, but now is everything become a hydrogels like this. So this is like a solid. See, then can show that it's really like a as you can see that this is like a solid, and then but you see the textures of it. They look like a gel, like this. So then these are packing into like the situations. So maybe you can much more easier understanding the situations right here. So through this you can put the proteins now. So which is really well packaged system, as you see in the real example. Then the proteins are in here. So then there's a pressures. There's a pressure goes in, so that proteins are actually forced to travel. So they force to travel because of the pressures goes in. And then to which you have a different fate. The first scenario and the second scenario, which is a fate, is that some proteins are going into this side, into the bead. So we can imagine these beads, individual beads, are like the second case of the previous our class, which is the agar system. So then these polysaccharides in here in these beads are entangled physically to each other to make a networks inside. So individual beads has a networks in here. And then you can have proteins going. So proteins go inside and travel through these beads, the networks of the polysaccharides, and then they travel for a long time. The other cases is that the protein has a little bigger in size. Actually, they cannot go in because of there's a network has a certain size, and then they actually travel between the beads like this. But if there's a size in between, also the important concept is distribution of pore, which is, as I mentioned before, even the size of individual pore is a different size, right? So that some proteins can go inside, but can be excluded, and go inside can be excluded, right? So then you have scenarios that small protein always travel through the pore, through the pores, right? But the second case is that I explained before. Second case is in between beads, and also, but in some cases they can go through the pore, right? But the third case is a very obvious that it's a really big proteins. Never ever goes into the pore. So then always always between the bead. Between the beads. So then you can imagine now, as size of the protein being bigger and bigger, actually they goes rather than between the bead travel pathway, rather than they actually go inside the beads and pore traveling through this pore networks. So once these proteins are always travel through the pore, then if you think about the time, then the time requires to travel from here to here in a certain distance that has the longest, I should say, the longest time. The longest time required. But this is in the middle, the second. This is the always between the beads means the fastest time travel throughout this through this. So this now giving the very clear ideas for you to imagine now, right? So you can have, say, the beads has certain networks up here inside. So the proteins, say, big proteins, middle protein, and small proteins that, say, the times actually the longest. So I'll put this number 1 and this 2 and 3rd. So the illusion time that we define, that when we think about time 0, that when pour these proteins into the hydrogel, time 0, and then the illusion time means when these proteins are traveling whatever their pathway and then going into the bottom of this big column. The time is actually now, you will see if there's 3 cases, then this is the longest one, so that it travels longest one, so that this will be loading time much later on compared to this 3 and so this 1 and this is maybe 2, this is maybe 3, for example. Right? So, but you have to think about different way. So as I mentioned before, this is the cases I show in the real demonstration was Cepheid X, G75. The 75 means something indicating for the pore size. So that there's a G50, G100, and G whatever 25, something like this. So that actually giving you different pore size, for example. So some G larger value, then providing the larger pore size like this, so that you can have a resolutions of this. Even proteins comes bigger, or even protein comes much smaller than this, depending on the size of the beads. You can have a range of the separations like this, either or this range or that range of the beads. You can put them separating, because if pore is bigger, then probably these smaller things always travel, and they can make a resolutions in a very nice way. But if you can put like this, then this cannot go in, this cannot go into the bead, so they have to always travel through between the beads, so they can actually traveling time is much faster. But the two different proteins are two different things, but actually eluting the same, in the time. That's why it's very important to actually choose which beads that is a different pore size are packed in here, and then that range of the proteins is separating. In other words, excluding not going into the bead, extruding out from the beads so it can separate the sides. So we talk about the gel permeation and side exclusion chromatography in a briefly way. So we will continue the next one for a little bit, touch more about the gel permeation chromatography a little more, and then we go into the next topic, which is the ion exchange chromatography next time. Okay, thank you very much.